Hello and welcome to Science Research Weekly. This is Season 5, Episode 2, Beat AI by Being Better Than Average. Get ready for supermassive dark stars, human creativity versus AI, unsinkable metal tubes, robot-assisted microscopy, the collaborative Microbial Metabolite Center knowledge base, and a grant to strengthen agricultural systems. This will cover material from January 23rd to the 29th. Let's get started with some research articles. First up, we have supermassive dark stars and their remnants as a possible solution to 3 recent cosmic dawn puzzles. So researchers were looking at these puzzles that are coming out from data from the James Webb Space Telescope. The first 2 of the puzzles are the presence of blue monsters, ultra-compact, very bright, high-Z galaxies, and little red dots, very compact, dustless, strong, Balmer break cosmic dawn sources. So models of cosmology, those observations break them. There's also a further problem, I think, with the origin of supermassive black holes in the most distant quasars. So the authors posit a theory of actually dark stars, but not just dark stars, supermassive dark stars, so they're made of powered by dark matter annihilation. And these supermassive dark stars would offer solutions to those 3 puzzles. So that's what they go over in this article. Next up, the article Divergent Creativity in Humans and Large Language Models. So they essentially took large language models and tested them for creativity using this thing called the Divergent Association Task across different creative writing tasks. And they pitted them against, I think, 10,000 humans. And while they did find that overall, GPT-4, there's a bunch of different LLMs, GPT-4 did better overall than all humans. Turns out if you split humans by sort of levels of creativity, the top 50% did better than all the other models, and even more so for the top 25 and the top 10%, which sort of means that the most creative humans, and actually the average human really isn't creative at all when it comes to like technical terms of creativity, of creative output. So it looks like if you're in any way creative, better than 50%, you do better than a large language model. So it kind of shows that there might be an upper limit of how well these models can sort of regurgitate creativity. Certainly an interesting actual benchmarking of creativity against humans. So for our third article, we have Dripping to Destruction, exploring salt-driven viscous surface convergence in Europa's icy shell. Europa is a Jovian moon, has a liquid interior, might have life, but it's unclear if oxidants from the surface could actually get into the interior, and the authors actually look at a potential mechanism called viscous surface convergence. And so they go through how that might happen to get, again, material from the surface into the interior of the moon. Final article, published article, in geometry-enabled recoverable floating superhydrophobic metallic tubes. These are these tubes that essentially are unsinkable, that because of their structure and how they repel water, they actually can't be sunk even if they sort of are pushed underwater. And so developing these could lead to incorporating them into marine vessels to keep them from sinking no matter what. So really interesting sort of engineering work here. All right, let's switch over to some preprints. Let's start with archive. We have the first one, extreme winds on the emerging dayside of ultra-hot Jupiter. So using spectrometer data from the Keck Planet Finder, looking at the ultra-hot Jupiter Kelt 9b, looking at potential sort of atmospheric movement and content. Next up, we have how well is the local large-scale structure of the universe known, CosmicFlows versus Biteau's galaxy catalog with cloning. So to look at the actual density distribution of matter in the local universe, they compared a couple different methods with pros and cons for each. And then the third one in archive, we have learning from a steady hand, a weakly supervised agent for robot assistance under microscopy. So if you've ever done really fine microscopy work, sometimes it's hard to like move around a tip to get to the right position. So it turns out you can use like a robot arm to help you with that. And this paper looks at a weakly supervised framework to improve that support. All right, switching over to bioRxiv, I got 3 articles. The first one is Community Curation of Microbial Metabolites Enables Biological Insights of Metabolomics Data. So a bunch of authors here are trying to bring together microbial metabolomic data into something they call the Collaborative Microbial Metabolite Center Knowledge Base, or CMMCKB. Also on bioRxiv, there is the preprint, a genetically encoded cysteine biosensor to monitor cysteine dynamics across life domains. So cysteine is a central metabolite. It's really useful, but detecting it is hard, especially in living systems. So the authors have a genetically encoded radiometric green fluorescent biosensor for cysteine. And then finally, we have the preprint, unifying the genetic landscape of common and rare diseases via related neighborhoods. The authors use a deep learning method that couples a variential autoencoder to network embedding to essentially be able to map the landscape of human diseases, taking in data from clinical reports, genome-wide association studies, mouse phenotypes, and gene oncology to really map, how diseases work, especially how rare diseases might be related to common diseases. To look at mechanism and be able to actually look into experimental models, targets, drugs, whatever, to actually solve for those diseases. All right. Let's see, just have a little bit left here. Some new are CRAN packages that might be of use. There is 3. There's GLM Starma, a double generalized linear models for spatial temporal data. There's RepFun, create tables, listings, and figures using functions styled after SAS macros. So that might be helpful if you're moving from SAS to R, or you know how to use both. And then finally, there's Give Me My Stats, statistics utilities. And then, closing our time here together, we have one grant from grants.gov, Agriculture and Food Research Initiative, Strengthening Agricultural Systems. So this is from the National Institute of Food and Agriculture, looking to strengthen agricultural production while enhancing farmer prosperity. Some of the sort of specific pieces or sub-priorities that would go into an application would be new uses and expanding markets for agriculture and forestry products, solutions to pests and diseases of plants or animals, and combating food and dietary related chronic diseases. With that, thanks for joining me for Science Research Weekly, and as always, science on.